Hello, hello again. Niaz Mohammed is here to talk with you with environmental microbiology and public health. What is environmental microbiology? We are talking about the bioremediation and bioremediation research. This is a very big domain because bioremediation is getting more and more interest on this globe. It has two reasons. It has some reasons because population on globe is going to increase. When population is going to increase, the demand and the requirement are more required for the more public. They are producing more waste. They are using more and more things. Those are converted into waste material. So load of waste material, especially xenobiotic compound concentration and content, is gradually going to increase on this globe. Therefore, if someone will use chemical and other techniques to treat this pollutant, will be more and more expensive. And that also can cause most secondary uh, effects on this globe as well as on human health, animal health, environmental health. So most of the people are going to direct bioremediation. And all cities, mostly all developing countries and developed countries, they are relying and as well as they are proliferating, as well as they are suggesting bioremediation to treat the organic and inorganic pollutants on the globe. Because bioremediation have no secondary consequences and problems, and it is naturally going phenomena for the cleaning of globe to maintain the life on globe. So here we have to talk bio-augmentation versus bio-stimulation. What is bio-augmentation and what is bio-stimulation? What is difference between these two techniques? Biostimulation involves the modification of environment to stimulate existing microorganisms capable of bioremediation. So in this title and in this heading we have talked too much. Biostimulation involves the modification of the environment by supplying selective nutrients and by providing selective environment to stimulate the existing microbial population with potential to degrade to decompose that particular content, pollution content, to stimulate existing microorganisms capable of bioremediation. Just what we are doing, we are providing nutrients and we are providing conditions to stimulate the existing microbial populations which have the ability to for bioremediation of that particular content. Endogenous population may not be capable of degrading the xenobiotic or the wide range of potential substrates present in complex pollutant mixture. So this is the issue. Suppose there is a place where pollution is occurring, okay, where any spill or accident of pollution takes place. Maybe the microbial populations which is there may not be able to degrade the xenobiotic pollutant or the wide range of potential substrates or that pollutant have many more compounds and the existing population may not have the potential to this mixture of pollutants. This may be a problem. So what we have to do for this? Endogenous population may not be capable of degrading the xenobiotics or the wide range of potential substrates present in complex pollutant mixture. This may be because some soils are enriched for the microbial communities. They have many more microbial populations. From them, some microbial populations are capable or have ability to decompose the various type of pollutants. But some environmental conditions and needs are there which have less microbial populations, which have specific microbial populations. Maybe they not have ability to degrade the various type of pollutants. Suppose desert area. In desert area, if some pollution is going on, if some accident of pollutant takes place in a desert area, what is problem with desert area? In desert area there are less microbial communities due to extreme harsh environment and due to less nutrients, and microbial populations whatsoever are existing over there, maybe they have no potential to degrade that various mixture of those pollutants. So here there is a problem. So what we are doing in bioaugmentation is the introduction of a group of natural microbial strains or genetically engineered variant to treat the contaminated soil or water. So we concentrate over here. Bioaugmentation is the introduction of addition of a group of natural microbial strains are a genetically engineered variant to treat contaminated soil or water. So in bioaugmentation, what we are doing? We are introducing new microbial population or any genetically engineered microbial population to decompose that pollutant. That is called bioaugmentation. So current research is going on for bioaugmentation because we have to develop new bacterial strains which may be environmentally friendly, which should be safe for human, generally considered as safe. Gross, we can say. Gross bacterial strains we can produce, we can prepare in laboratory and can use for beneficial purpose to clean the environment for greater human interest. But still we are afraid that maybe this bacterial strain will cause any secondary consequence. So biostimulation is to stimulate the existing microbial populations with capability of the bioremediation by modifying the environment through addition of nutrients and air. But in some regions, in some areas where pollution will take place, the microbial populations, interior microbial populations of that area have no ability to decompose the various type of pollutants. Therefore, we must have to introduce any new microbial group or genetically engineered bacterial group to treat that particular pollutant in from environment. So that is called bioaugmentation. If we will study the bioremediation, what we can do, where we can get jobs. So there are many domains. After reading this bioremediation, we can work. Our services can be hired or required for the globe because globe is the actual asset. If that is in a balanced form, life may exist for longer time. If this globe will be full of the pollution, so life it will be difficult for the life and living organism. So as a career opportunities, we can be outdoor inspectors where we can visit to the environmental sites, uh, accidents where pollution is present or pollution occurred, where we can go, we can examine, we can collect samples, we can report, and we can protect that site. In another side, we can work in a laboratory to check the type of pollutants. Okay, various types of pollutants are coming from the environment, from the field, and we can analyze the potential and chemistry of the pollutant to decide that which type of strategy can be adopted for this treatment. We can work on administrative services to design and to control the whole chain to care and to manage the bioremediation. Government, maybe government employees, regulatory, our sites can work in any company. So there are many domains after completion of the bioremediation work in environmental site we can do. So in summary, many factors control biodegradability the degradability of the contaminant in the environment. So from first, if you will see this thing, there are many factors which can control the elemental cycling or bioremediation, or biodegradability or biodecomposition of different contaminants in the environment. Suppose pH, temperature, oxygen concentration, nutrient concentration, environment, moisture content. These all factors are influencing the biodegradability, bioremediation, or elemental cycling of the contaminants in the environment. Before attempting to employ bioremediation technology, one needs to conduct a thorough characterization of environment where the contaminant exists, including the microbiology. So we cannot apply any strategy to remove the pollutant from the environment. So what we have to do? We must have to do characterize the whole environment where pollution is present for adopting a strategy. To adopt a strategy where the contaminant exists, including the microbiology, which type of microbial community is there? Is that microbial community have ability to decompose that particular pollutant in that environment or not? If yes, it is okay. We have to increase the microbial population by providing nutrients and the concentration of oxygen and other things and conditions. We can improve that microbial population. We can boost, we can stimulate that microbial population by providing nutrients and conditions. If not, what we have to do? We have to tie our hands? No, not at all. We have to introduce bioaugmentation. We have to introduce new microbial populations and strains from environment or from laboratory which has been genetically engineered or naturally adapted microbial population to that particular environment for the for the degradation of that pollutant. Must have to check geochemistry that which type of soil is present over there, which type of components can react with the pollutants which are occurring over there. What is mineralogy status? Geophysics, what is the soil structure? It can move towards downward or not. Hydrology of the system. So after analyzing all these characteristics, we can decide and we can implement any strategy for the any strategy of the bioremediation to decompose, to degrade the pollutant. So it is overall about elemental cycling and bioremediation. This chapter is our now, which we have started from uh, elemental cycling. We have studied elemental cycling on uh, terrestrial regions. We have studied elemental cycle in water bodies. We have studied applied sites of elemental cycling where we have treated potable and drinking water, as well as we have treated and converted components which are present in wastewater to make it clean for the environment. We have studied pollutant types, its nature, its classifications, uh, rapidly degradable, somehow degradable pollutant, rapidly degradable pollutant, non-degradable pollutant, and recalcitrant pollutant. And we have calculated that 90% of the pollutants those have organic nature and can be decomposed easily by means of microorganisms. In second phase, we went to the bioremediation phenomena which is going on nature, uh, and from the history of this globe, microbes are responsible to maintain the uh, life on Earth by cleaning pollutants, by providing elements for the new growing life in soil, especially primary producers. Microorganisms are playing a vital role because they are decomposing all waste material and converting it into elemental form, which are very essential for the life. And secondly, microbes are providing fertilization for the better growth of the environment. But sometimes if these microorganisms which are present in our environment, whenever they will go to human body by any means, especially by water, they can cause various diseases. We have discussed those diseases also that can increase our burden, financial, economical, and social burden on human societies. So for that, what we did, we have treated the water in a best way to provide good quality of water to its public. And second, and another thing, if you will go ahead of this, bioremediation is natural going phenomena, but sometimes uh, it is going to be applied because in natural attenuation system, our natural bioremediation is very slow due to various factors, and sometimes we want to stimulate it because that is the area of the public interest where people will go, human animal activities, live activities are taking place, so it is danger that can cause any hazard to the public. Therefore we are trying to make it faster and faster by bioaugmentation, biostimulation. Some other techniques also we can use for the cleaning of the environment. Those are air sparging system and phytoremediation system. Phytoremediation is also a technique of the bioremediation. What we are doing actually in phytoremediation technique, in phytoremediation, we are selecting the contaminated soil, we are growing plants, and what will be next? And plant will absorb pollutant inside of it, and it will be fixed for longer time in environment. So you must have to study elemental cycling, pollutant types, bioremediation types, and in exam uh, questions will be asked from here. So I hope you will understand something little bit from here, but not too much. For to understand completely, you must have to go through books, through literature, through research articles, which has been provided to you, as well as these are available in laboratory, sorry, in library, and you can download it from internet. And some guides and some points we have explained in PPT also. So thank you very much. Have a nice time, please.